Hi guys, so we have a very short learning unit here on membrane transport, and one of the things that we did at the beginning of the chapter was kind of relate our four concepts of information, energy, catalysis, and structure. Same four things that we applied to our macronutrients. We sort of applied this to membranes and thinking about how they could affect that. So from an information standpoint, membranes do have a inside and outside sort of layer. So we can have information depending on what's displayed on the inside and the outside of the cell. We talked about that in our carbohydrate chapter when we talked about our blood group chemistry. And in terms of energy, this is kind of a big player here. Because membranes are used to separate the inside and the outside of the cell, we can build up concentration gradients which allow us to harness thermodynamic driving potential to do work. And so we're going to talk about some of those in this chapter. In terms of catalysis, even though these membranes don't participate directly in catalysis, they indirectly facilitate catalysis by allowing for the compartmentalization or the segregation of different metabolites. So by controlling what is where, that plays a role in catalysis. And then obviously structure. Structure is really important in a component of membranes in that they create the structure. So the ability to have these compartmentalized areas with eukaryotic cells is really important. So once we kind of talked about those general points, we moved into some equations that allowed us to think about the thermodynamics of a transport. And again, no equations that you need to know, but just the understanding that when you build up a concentration of one species on the outside of a cell that's different from the concentration on the inside of the cell, just by our delta G equals minus RT ln of KEQ equation, and the equilibrium being the movement of that species across that barrier. The greater the difference in concentration, the greater the thermodynamic driving potential. And with that, if that molecule is charged, we have an additional component. So that was those two equations that we talked about. Not equations that you need to know, but to understand that the buildup of a concentration gradient contributes to thermodynamic driving potential. We then had a diagram that we went through that allowed us to think about how we categorize different types of transport mechanisms. Whether they're mediated or not has to do with whether a molecule is hydrophilic or not. And then whether removing a concentration or a species with or against its concentration gradient has to do with whether it's facilitated diffusion or active transport. And then secondarily to that with active transport has to do with what's the cost of the money for moving that species against its concentration gradient, and then whether it's a cationic or a neutral species. So again, that sheet kind of walks through the details of those. We then spent some time going through and talking about different kinds of transport mechanisms. So I'm not going to go through the details in this little overview here. But again, we talked about things like 2,4-dinitrophenol, that diet pill, that allows us to move protons across a barrier. We talked about valinomycin, so that antibiotic that allows us to move potassium ions across bacterial cell walls, and that again is lethal to bacteria. We then talked about, in bacteria again, how they get starches as food inside their cells. So we talked about porins. And then we spent a good bit of time talking about the potassium channel, and that's really important because people couldn't figure out for the longest time how you could let the bigger potassium ion in but not the smaller sodium ion. So we kind of revisited some thermodynamics to think about the cost of dehydrating an ion in order to get it to pass through. Looked at some structural information with that protein, so a lot we kind of talked about with that protein. And then at the very end, we kind of talked about glucose transporters, and then we put it all together with thinking about how you get both glucose and sodium out of your gastrointestinal tract and your small intestine, and the importance of kind of having those two species together in your intestinal tract. So starting from the very big sort of why do we need to think about membrane transport to some very specific examples. Short, brief learning unit on membrane transport.